Good morning. I'm Marty Downs. I coordinate the LTER network office. I'm going to take just a few minutes to go over what LTER site exchanges are, what kinds of projects we encourage, and what the application entails. Then we'll hear from 3 of our 2025 recipients about the projects that they carried out. Site exchanges are intended to support the kinds of intensive, hands-on interactions that simply can't happen over Zoom. The 2025 projects focused mostly on methods, and those are always welcome, but we encourage you to consider other kinds of projects as well. Those might include intensive mentoring, project development, or job shadowing, but I'm sure there are lots of other types of projects that we haven't anticipated. The LTER Network Office pays directly or reimburses Site Exchange Fellows for all the travel-related expenses, including air or ground transportation, food, and lodging. Where needed, we've also covered expenses for field assistance, but we do not cover salary. Any LTER participant is eligible, including investigators, students, and staff. We'll also consider applications from researchers who are not currently affiliated with LTER, but who have an idea that would mutually benefit themselves and the LTER network. Site exchange applications are due March 31st. To prepare a compelling application, you'll want to start now to identify a collaborator at another site. Talk with them about the work you are interested in and listen to what excites them. Learn about some of the logistics at the proposed site. Where can you stay? Do you need to make special plans for field work or boat access? If you're a student or a staff member, talk to your supervisor early to be sure they're supportive. We'll send an automated email when your application goes in, so you want to have that conversation early. And if you aren't sure if a project seems right, please don't hesitate to reach out. So now we'll hear from 3 of last year's Site Exchange Fellows. Mingyu Zhang went from Plum Island LTER to the Virginia Coast Reserve. Alexandra Cabanelas Bermudez went from the Northeast Shelf to the California Current LTER. And Margaret Baker, a PhD student at the California Current LTER, went to the Northern Gulf of Alaska. I hope their work inspires you and helps convince you to take advantage of this exceptional opportunity.
All right. Hi, everyone. I'm Mingyu Zhang. I'm a current second-year PhD student at the Yale School of Environment. I work with Professor Peter Raymond, and I'm very happy to be talking today about my site exchange experience. So a little bit about myself is I'm very interested in the lateral exchange of inorganic carbon and greenhouse gases from blue carbon ecosystem, so like salt marsh, mangrove, and seagrass. I have experience, I'm based at Plum Island LTER, which is one of the largest mangrove salt marsh site at the northeastern United States. And also my lab has done some very extensive field work at the Florida Everglades site, which I joined for a couple of times. So I thought, well, I have experience working at both salt marsh and mangrove. It would be really interesting to go to another seagrass site to complete this blue carbon map. So I'm also interested in looking at process-based models to quantify these processes at scale. So as I keep on working in these ecosystem, I noticed there are some pretty consistent limitation in the field. First, that there is very limited direct quantification of inorganic carbon greenhouse gas fluxes. We have snapshot sampling, but there's not enough coordinated process-aware data sets. And also there's very limited pore water biogeochemical data in blue carbon ecosystem. And that's a big issue because pore water is where a lot of the chemistry I'm interested really happens, like sulfate reduction, iron cycling, carbonate dissolution. And these processes generate alkalinity and methane. So without the pore water constraints, it's really hard to validate our understanding and also validate the process-based models. So third, there's very limited linkage between the sediment process and surface water or atmospheric flux measurements. So I want to use the tool and sampling method I have that is already pretty fine built for salt marsh ecosystem and migrate them into seagrass bed and see what interesting outcomes I would get from these ecosystems. So timeline wise, after I have built my big picture ideas, the first step was really figuring out what's the research question that's both interesting and also feasible within a week. It's very easy to propose something very ambitious, but the key is making sure it can be realistically done within the time frame and site logistic and the people available there. So the second would be to reach out early to the LTR community that you're interested in, like the site coordinator, active students, and PI. That part was really interesting to help me refine my idea and narrow down my research plan. So after you get to know what instruments are available, what's realistic for the field deployment, what's the housing situation like, you can write up the field proposal and make a budget plan. One thing I found really helpful in writing my proposal is to always keep in mind, like, how can your research be actually helpful for the exchange site? And why is traveling to that place actually necessary? You don't want to step on other people's toes. And yeah. So while I was doing background research on through the Virginia Coast Reserve LTER community, I was really fortunate to be connected with Dr. Peter Berg and his student Paola. Peter's group has a very interesting and unique way of measuring gas exchange velocity through this upside down eddy covariance system. Gas exchange velocity is honestly one of the parameter everyone use, but it's notoriously tricky to quantify accurately, especially in estuary ecosystem. So in our lab, we have a more traditional approach, which is shown in the picture on the left bottom here, is using a bucket or a chamber to measure the fluxes directly. So once we connected, it felt like a really natural collaboration to compare method at the same site under the same condition. So that this conversation happened kind of organically after I started looking into the VCR community. So basically I made 3 plans. The first plan is to look at alkalinity and also measure greenhouse gas fluxes using chamber system and cross-compare it with Dr. Peter Berg's system. And I picked the site where Dr. Berg's group usually do his time series analysis. So I just followed his sampling spot. And then another is to look at the surface water transect, which is also a long-term monitored transect carried out by the VCR community. So I picked their spot and it just added additional surface water quality parameter I want to measure, like GHG, greenhouse gas emission, alkalinity, DIC, and also isotope across these sites. And then the third is one of the most interesting thing I looked at. It was pore water redox at the seagrass and marsh bed. So I was measuring redox potential and collecting pore water at these sites. This is, so you can see here, I made my schedule pretty compact, but so on paper, everything looked very well organized and efficient. But the thing is, like, the weather won't cooperate, tides sometimes don't line up perfectly, and instruments take longer to deploy than you think. I would honestly say whatever time you think something will take, multiply it by 3. It's just field work always takes longer than expected. So I didn't get everything done exactly as planned outlined here, but that's okay. Some components worked really great and some had to be shortened and some evolved once I was there. So it's just interesting to see how things evolve once you're actually at the site. So next are just very fun pictures of us sampling at the site. This is us on the transit cruises and also deploying the ISCO to sample the surface water for a time series analysis. And this is the gas chamber measurement to measure gas exchange velocity. So you can see on the right here, this is Dr. Petersburg, very sophisticated instrument. And this is our simple tool to just a chamber and a bucket floating on the water. And then also pore water sampling. Actually, I have to be honest, seagrass bed is one of the most enjoyable field sampling I've ever done. It's better than the mangrove and salt marshes. And so right now I'm going through I've already measured all my sample. I took them back to Yale for analysis and going through the data analysis part. This is just a brief overview of what the transect data look like. It's quite interesting. You could see there are spatial variation for the methane concentration and methane isotope. So I'm waiting to do more data analysis on these data. This is a quick overview of the pore water redox profile, comparison of marsh and seagrass. It's very interesting that the seagrass bed are mostly deoxygenated, whereas marsh, you still get some oxygen flush into the sediment, whereas seagrass is basically out of oxygen. So looking at the expected results, I will be collaborating with Dr. Petersburg group. Sorry, my leg just. Dr. Petersburg's group on a gas exchange method paper. We have some very interesting results that's coming up, so I think we'll work on that towards a manuscript. And the pore water data will be used to cross-compare with marsh and mangrove sites. That's ongoing effort and to constrain a modeling component. And the transit data will be further analyzed through like PCA to look at spatial patterns. And I know VCR has done the transit water environmental data collection on a monthly basis. So it'll be interesting to look at that in a long-term monitoring situation. So I like to wrap up with an acknowledgement. Very grateful to work with Peter Berg and his student Paola, and also I like to thank my advisor for giving me the opportunity to travel, and also the Beck who helped me throughout the way, travel with me to the site and helped with a lot of the sample collection, and also the VCR LTER group, which is a really warm, welcoming team over there. I loved working over at that site. And last but not least, the Exchange Fellowship and Marty. Yeah. Thank you. Marty, I think you're muted.
Any quick questions for Mingyu? All right. Well, then I think Marguerite is not with us, so we'll have to hear that at some other time, but we'll move on to Alex.
All right, I think you can see my screen now. Okay, let me move this out of the way. Hello, everyone. I'm Alexandra Cabanelas. I'm a PhD student at Woods Hole Oceanographic Institution. I'm part of the Northeast Shelf LTER, and I was very fortunate to visit the California Current ecosystem so I went from Massachusetts East Coast to the West Coast. And so I'm excited to tell you a bit more about my experience. So I figured I would start with a brief kind of overview of where I'm coming from and what we do at our site. So we're located on the Northeast US Shelf. It's a very productive continental shelf, and it's very seasonal. So seasonality is very important to us. And so we go out 4 times a year, once for each season, to sample the plankton, from very small bacteria to large mesozooplankton and small fish. And you can see on the right side of the map, you can see our transect. So we kind of leave from the Cape Cod area, and then we have stations that we sample out to the shelf break. And then on the left map, you can see kind of the bigger overall picture of the entire continental shelf, which we have collaborations with NOAA to get additional data from other regions of the shelf. And so in our project, we're really interested in better understanding planktonic food webs, how these change across space and time, and how they're influenced by disturbances and changes to our ecosystem. And where I come into this project and what I'm interested from a research perspective is really on the zooplankton, food web ecology and zooplankton ecology. I also study forage fishes, which are fishes that eat zooplankton, but for this, I'll be focusing on the zooplankton aspect of my work. And so one of the ways we sample zooplankton, and what I'll be focusing on for this, is we use bongo nets. So these are 2 nets that are kind of connected. They're of different mesh sizes, but I'll be focusing on samples that come from the 150 micro mesh net. Here's some nice pictures of us sampling at night, during the day, any time of the day. We put these nets in the water. And so from this, we get really good composition data. So we know who's out there, when, how many, and it's very nice because now we've been sampling out since 2018. So we almost have a decade of kind of data on the plankton. However, our big gap right now is that we have limited size information. So we currently don't measure organisms or get weights. So we really lacking on that aspect of the food web. And it's just size is so important for flora and fauna in any type of ecosystem. And it really affects how the food web is structured. So this was kind of the main gap I was seeing in our project, and something that would contribute to my thesis, and also I think improve our understanding of our system. So that's how I went into the site exchange application. Like, okay, I think it would be really important and relevant to get size information on our zooplankton. How can I do this, and where could I go? So this turns into YCCE. So here's a map of the 4 LTER sites. Sorry, Moria, I didn't include you here, but yeah, there's Moria coral reefs too. And so this is kind of what I first went to, but I also wanted to clarify that I might as well pick the lake ecosystem or a river. I think there's a lot of value in visiting a different ecosystem. So I just want to highlight that if you're marine, you don't have to go to marine site, and vice versa. But so I looked at this and I was like, okay, CCE is one of the older ones. I would have loved to go to Antarctica, but the logistics would have been a bit more complicated. So CCE seemed like a great choice. They were established in 2004, and they just have really, really great and established methods and protocols for this type of work. Additionally, CCE has 2 amazing PIs, Moira Decima and Mark Oman, who I got to work with, and they just have a lot of expertise in zooplankton ecology and food webs. And also they work on other aspects like stable isotopes, which are very relevant to my thesis work. So with this, I identify CCE as a good fit to possibly answer this question and get this data for size information. And so, and ultimately, they also have this instrument, which I'll explain a bit more what it does in a little bit, but they have this ZooScan instrument, which we currently have here at Hawaii in my lab, but nobody knew how to use. So it's just been sitting there. So I was like, okay, this is great. Both Mark and Moira have ZooScan. They use it almost daily. This would be a great point to go out there, learn, and then come back to Hawaii and keep scanning my samples. And so I had identified, okay, CCE works. They have the equipment. They have the lab set up to answer my question. And then I also wanted to mention a little bit more background about how I made this happen. I actually had met Mark and Moira about 2 years before the site exchange. I was able to participate in a synthesis working group at NC's where I got to meet them. So I just wanted to highlight that there are other opportunities within the network that can eventually lead to a site exchange or some other type of collaboration. And the synthesis working group also let me see, we were working the 4 LTER sites that I showed previously. The 4 marine sites were part of this working group, so I was able to see what types of methods were very similar and where we could maybe improve and join forces to have more similar data sets to then allow further synthesis work to happen in the future. So that was another thing I noticed. Alaska and I think even Palmer, they have image data for plankton. So that was something that maybe could strengthen the NES if we had that as well. And I also wanted to highlight that I actually was submitting my application while I was at the ASLO conference last year. And I had the opportunity to meet Moira's PhD students and talk to them in person about their research and just make sure that it was a good fit for me to visit their labs. So I just wanted to give that kind of extra context. And so going back to what exactly I did, so like I mentioned, there's this instrument called the ZooScan. It kind of works like a regular scanner. On the right hand, you can see, you can open it up and it has a basin where you can pour your plankton sample and then you close it and you can get a digitized image of your sample. And so that was my main goal to visiting CCE, was to learn how to use this, learn their workflows, and learn more about the machine learning applications they're using to process the images. And so a little bit more about how this works exactly. Here's a real picture of my site exchange. This is the ZooScan at Scripps. And on the right side, you can see I've already poured my sample, and you see all those little dots are little tiny Coke pods. And then usually spend between 30 minutes to an hour physically separating all the organisms so nothing's touching. And so there's a picture of kind of the process. This is a nice one with the larger organisms, so it's a bit quicker, but for the little ones, it does take a while. Oh, and I should highlight, I actually sent samples from the Northeast US Shelf to Scripps and was able to process NES samples while I was there, which I think was very valuable. And so this is kind of what it looks like, that you can digitize the sample, and then it kind of creates this like nice image of everything. And then you can extract individual vignettes and get really awesome digitized information about your sample. And you can get the size information, but you can also get to shape information about kind of transparency. You can also see other things that are harder to see or to get information from just our abundance data. So on the lower left, you can see the copepods with the eggs. So, which is often missed by just looking at the raw abundances numbers. And then you can see kind of other features, like maybe on the top right, this is an arrowworm. You can see the little kind of bristles on their head. So it's very awesome because you can get more detailed information about the community, not only who's there, their sizes, and maybe other details that you might miss from the other data that we have. So here are some extra photos. I took samples from summer. I was able to process 9 samples while I was there. And so in the summer, we get influx from the Gulf Stream water into the continental shelf, and we see a lot of crustacean larvae, which are very cool looking. And all these organisms are extremely important for the food web. So it's really information that will help us improve understanding of the system. And so I just also wanted to end with other things I did beyond scanning. All the PIs I worked with, including my advisor, were very encouraging of don't just spend the 2 weeks in the lab scanning, try to do other things. So I was very lucky. I was able to attend a couple of ecology seminars that were actually very relevant to my work. Moira also had another visiting student that was also ZooScanning, so I got to interact with her. She was visiting from New Zealand. I was able to participate in lab meetings and give a presentation about my thesis. So I was able to also talk and interact with scientists about other chapters of my thesis beyond the ZooScan work. And I also learned about other methods. So they showed me how they get dry mass and biomass in the lab from zooplankton samples similar to ours. And it was just truly such a great experience. Initially, we planned to do this in August, and then we had to switch it to December, which was actually very happy for me because I got to escape the cold in Massachusetts and go to the beach in California, which was amazing. And with that, I would like to end saying thank you to Moira and Mark, to Oceana, which was a technician who trained me. She met with me on a Sunday at 7 a.m., Sunday after Thanksgiving. So I'm very grateful to all her help, Dante and Grace, and just everybody else. It's kind of Christmas time, so there were a lot of holiday events going on, and I just felt very welcome. So I'm very grateful for the network, for this opportunity, and Marty for always organizing these cool opportunities for and also thank you to my advisor for being so supportive to this type of activity. And yeah, happy to answer any questions. If you ever think you would like to visit or are interested in what other types of work we do at the NES, I'm happy to chat and answer any questions.
Thanks so much, Alex. So is the ZooScan instrument at Hui working now and being actively used?
Yeah, it is working now, almost being actively used. We're getting a new computer because our old computer was just way too old. But it is working. And we also have another piece of instrument called the flow cam. So I'm actually working on using both and comparing how those complement each other.
Oh, that's cool.
Oh, and actually, I wanted to highlight just very quickly, the grad students are hosting a Flash Talk Friday. I'll share the link again in the chat, but it'll be very casual and just another great way for all of us to learn about each other's work. So if you're interested, sign up. I'll share the link in the chat.
Awesome. Thanks for the reminder. Any other questions for Alex? Okay. Well, we will move on to Maggie.
All right, give me 1 sec to refigure out the screen sharing. This is not my strong suit. Okay, can you see my slide?
Yep, looks great.
Awesome possum. Well, let me move the little bar. Hi, everyone. My name is Maggie. I am super excited to be here today and share my experience with the LTER site exchange that I did this past year. So a bit about me, I am a third-year PhD candidate at Rice University in Houston, Texas, and I'm a member of the California Current LTER program. I have participated in the past 2 CCE LTER process cruises where I conduct deckboard incubations to investigate the effects of marine heat waves on primary producer community composition, physiology, and productivity. I adore my work on marine heat waves in the CCE, but my main thesis work actually involves Antarctic sea ice algae. So I am super excited about and interested in polar regions, which is really what led me to NGA when I started looking around at different sites for the fellowship. So a few weeks before the fellowship deadline, admittedly like 2 weeks before, I cut it a little bit close, which I don't recommend. I reached out to Tom Kelly at UAF, who I knew loosely through lab connections, and I asked if he'd be interested in an LTER exchange project. And he was all for it. And then the question became what would be the most beneficial as a project. And Tom was great and threw some ideas around before we settled on something more akin to intensive mentoring and skill building based off of a project that actually held a lot of interest for me, but I knew nothing about. And that was a bio-optical project, which is a project that Tom already had running, focused on 3 primary questions. Can we improve remote sensing retrievals of ocean color for our subpolar regions? What is the phytoplankton phenology for the Gulf of Alaska, and how is the ecosystem coupled to the spring bloom? And do these impacts extend all the way to fish? So for this project, among many other instruments that he works with, Tom takes a seabird eco-triplet and an ACS out to sea during NGA process cruises to try and answer these questions. In use, these instruments collect continuous data of things like particle backscatter, light absorption, and attenuation. And they're usually paired with a benchtop fast repetition rate fluorometer, and together these instruments can piece together a really nice picture of water column dynamics. So these instruments go out together to survey the NGA transect lines, collecting an exciting time series data set primarily focused on this Seward line here. And this region experiences really extreme seasonal variability that's characteristic of high latitude regions. So just a little more background on the instruments and the setup, because this was very relevant to what I did later in the project. On these cruises, these instruments are connected to the ship's inline flow-through system, so they're not deployed actively in the water. They are lab-based, and water from the flow-through system passes into a little debubbler, and then into the ACS secured in the sink to record light absorption and attenuation data. And then into this little black box, which is called a coffin, that holds the eco-triplet, which collects particle backscatter data. And the whole setup is valve controlled, and every 2 hours, the valves are supposed to automatically switch to funnel water through a series of filters ahead of the debubbler to automatically record blanks for baseline corrections. And this data is really valuable for remote sensing validation, which is especially important in regions like the NGA, which is influenced by things like glacial silt, salinity changes, ice cover, etc. And the data overall provides insight into physical properties of the water column, which can be used to characterize circulation, eddies, boundary zones, and inform on phytoplankton communities and particle abundance pertaining to carbon export. So up to this point, my experience was almost entirely made of lab experiments, and the data I work with is very, very small. Even at sea, I am doing deckboard incubations, and while I collect a ton of cool data, it's still considered small data sets. So I had not previously had exposure to working with large data and making those types of processing decisions. And also had no exposure to previous like bio-optical data sets at all. And while I do a lot of experimental lab work, I do hold a lot of value and interest in larger long-term data sets. So through this site exchange fellowship, I traveled to the University of Alaska Fairbanks to work with Tom in person and learn about the instruments and working with these large data sets, which involved quite a bit of programming that I also did not have any previous experience in. And the trip itself was all arranged by the L&O office, and I was able to stay in the UAF campus dorms and work on campus for the full week, which was really great. And it was beautiful. During the trip, I learned a lot about the instruments and bio-optical data, but it was kind of like a programming boot camp. I had some background in coding in R, and perhaps this is basic to some people who have been doing this forever, but I was able to learn a ton about large data management and programming. So basically in Alaska, I started off with coding, and I used data that had been collected from the ACS and eco-triplet previously on NGA process cruises to build a pipeline to transform the data and funnel it into a non-SQL database, MongoDB, for long-term and standardized storage. And the ultimate goal was to build a program to directly funnel data from the instruments, correct it, transform it, store it in a database, and then build out a pipeline to actually use it. Which leads me to the collaboration part of this program, where I really got my crash course in the instruments and even more programming. So like a month after my trip to UAF, I participated in the fall CCE LTER process cruise, and Tom shipped the 2 instruments and the full setup down from Alaska for me to use on the cruise in the CCE to develop a cross-site comparison of bio-optical properties. NGA and CCE are vastly different sites, with the CCE being at a much lower latitude. And one thing that we do differently from NGA is that we employ a Lagrangian method, where instead of hitting specific stations, each process cruise we actually pick a parcel of water and throw a GPS tracker in it and follow it for up to 5 days, which allows us to study a water body over time as it moves and as its properties change. So post UAF trip, Tom ended up completely redoing the system setup. And this is where I got more of a crash course in programming live time, where I set up the instruments, then had to take the untested rework system and work around the clock to fix bugs and get the system up and running. And I learned a lot about Python programming in like 3 days and got the system working. And please excuse how horrid I look in this photo, but this is the best picture I have of the new instrument interface right after it successfully connected for the first time. And basically, the new system completely bypasses the instrument-specific software. The data is collected and shuttled into a database format through a constantly running Python backend that's connected to an API controlling both the instrument status and a front-end user interface application that we can use to control things like the valve timing for the blank measurements. However, as with any new system under testing, there were lots of bugs. So I spent a lot of time on the cruise troubleshooting, and I'm still working with Tom to determine which data is usable. So please stay tuned for data and results from both the processing pipeline that we built and from the cross-site collaboration. It is an ongoing process to kind of wade through all of it. But in the future, one potential application of this is to also connect it with data collected by the ISIS DPI, an instrument that lives at NGA but also comes out to sea with the CCE. And the DPI takes really cool pictures of zooplankton in the water column, but it also collects data that the ACS and ecotriplet do. So we can eventually pair the two data streams together and unlock a greater understanding of water column characteristics in 3D. But regardless of the data, I learned some really valuable skills that I've already been able to apply to my own work. So outside of the CCE, I work on Antarctic sea ice algae physiology and survival over seasonal transitions. And I've used the programming skills that I learned through the Site Exchange Fellowship to develop a lighting system that directly mimics the ocean light spectrum and seasonal transitions and irradiance that are collected in the field from McMurdo Station, controlled through a Raspberry Pi. And I developed a front-end application connected through an API that allows me to monitor live time data streams and remotely adjust system parameters from my laptop, which might be rather basic for a natural programmer, but these skills have jump-started my ability to do a large portion of my dissertation work, and I know I'll be using them like daily throughout the rest of my career. So a huge thank you to the LTER network for supporting my site exchange. It was incredibly beneficial to me. And thank you to Tom and NGA for hosting me into my own site, CCE, for the opportunity to be involved in the LTER network and letting me take so much bench space at sea with these instruments.
That is really cool. Thank you, Maggie. There are so many different ways to benefit from these site exchanges, and it's not always clear when you start out what the major take home is going to be. And I love seeing the connection between the marine sites and the kind of potential for lining up methods. Does anyone, I'm guessing that a number of you are on the call because you might be interested in, well, so first, does anyone have questions for Maggie? Okay. Or any of the other speakers. I'm guessing a bunch of you might be on the call because you think you have an idea for a site exchange yourself, or you're interested in pursuing a site exchange. Does anybody want to throw out there what they're thinking about? It's a shy group.
I'm I'm currently looking around to think about PIs or technicians with experience in measuring root respiration, which is something that at our LTRs right now there aren't a ton of people doing. So,
And
but it's really
where are you coming from?
I'm at the Cedar Creek and also the Minneapolis-St. Paul LTR, so super close together. But thanks to all the speakers, it's really great to see like the range of ways that doing this kind of exchange can benefit everybody involved.
And I also want to highlight, Alex brought it up, but it doesn't have to be the same kind of site. There's lots to learn across ecosystems. That's one of the unusual qualities and one of the great strengths of the LTER network. So think about that as well.
Yeah, I did my master's in the Great Lakes, so I'm very passionate about marine and freshwater people interacting more.
Yep. There's so much to learn from each other that just gets buried in different literature streams that sometimes don't connect.
I have a clarifying question. So it seems like this is these types of exchanges, it's mostly for one person going from one LTR site to another. It's not necessarily like a full reciprocal, people switching sites or like going to one site together and then going to the other site together as well.
It doesn't have to be, but that is a potential proposal.
Okay.
So we certainly would consider uh, a, a true exchange.
Yeah, especially in the case of like doing like a distributed experiment where you go to one LTR site, you set it up, and you both go to the other LTR site and set it up in exactly the same way, right?
Yep, exactly.
Thanks.
I think it's a great opportunity for things like piloting the setup for a distributed experiment in a couple of different locations. It can be really easy to think in the abstract what's going to work in the location that you are most familiar with, and then you get into a location that's very different, and there are all sorts of considerations that just didn't, come up in your initial thinking. So yeah, that's a great.
Marty, just curiously, in that kind of setup, would two people at two different sites possibly like co-write a site exchange proposal together, or would they each submit a separate?
No, that would be ideal, would be to co-write it together so that, you're really clear that they are working together and on the same path. I should probably also clarify so the selection process typically is I'll do a first review of all the proposals, just kind of make sure that everybody's ducks are in order, that, PIs are aware and the person that you're proposing to visit and work with is on board with it, and then do a little summary of the proposals. The LTR executive board is the group that makes the final choice about which proposals are kind of the most long-term value to both the network and the individual. And the kind of criteria around that are really what's going to help move the network forward over time. So some of the factors that were most interesting in these proposals were what Maggie pointed out, kind of the opportunity for marine sites to compare and align their sampling methods over time, or the kind of transmission of more advanced sampling methods from one site to another. That's a big factor. I think with the growth of AI, interesting uses of AI or aligned uses of AI is going to be pretty relevant. But there are all kinds of things that are potentially of value. One one of the experiences that really inspired this program was actually an exchange of education and engagement folks that happened among 3 sites that were doing similar work. So the kind of driving motivation for it is what is going to benefit from this really intensive cross-site experience, from really spending time together. All right. Well, if we don't have any more questions, I won't keep folks, but really fun. Thank you to the 3 of you for your presentations. I'm so glad that you were able to get that much out of the experience.